I decided to extract my DNA at home. You put it back to the glass, then humanized me. In the way they felt. And somehow they produced and will contain these 3 billion letters. And out of dope. So I decided to go, you translate this stuff into letters. And they also reproduce quickly. In your opinion, what is the most important animal for human? A decade ago, Western media were alarming us about the world without the bees, which are crucial to our survival. And celebrities like Beyoncé or David Beckham suddenly started to produce bees and care about environment. Today though, bees are still around. Not like my girlfriend. In fact, the United States might have more honeybees than ever, with more than 1 million bee colonies added in the last 5 years, bringing the total to nearly 4 million. There is more bee colonies in the United States than people in Slovenia and Estonia together. The New York Times even called this hivewashing, referring to greenwashing, because it was just fake. Only wild species of bees were at risk. Well, this bee story was a bit out of scope. I think there are some animals that are as important as bees. It's rodents. Mice, rats, your friends when you go out. Do you know what Alzheimer, COVID-19, depression and lung conditions have in common? Mice explained us what this is and how to treat it. COVID-19 vaccines were developed at exceptional speed, and studies using mice were vital in this process. Even though mice cannot catch COVID naturally, they can be infected with the virus by creating genetically altered humanized mice. And I will talk about this in details later in this video. Today you can go mouse shopping and even choose a humanized mouse with the traits you want. So with this mouse, engineering scientists can pimp your mice exactly like Exhibit was pimping the rides.
How many girls you got to actually come party back here?
None.
Well, mice. According to FBR, Foundation of Biomedical Research, 95% of all lab animals are mice and rats. I must give you an important notice that the information may have a lot of variance in different sources. For example, European Animal Research Association estimated that in EU, including Norway, rats make only 52.5% of all animals in medical research. Nevertheless, all sources bring the same message. Rodents are indeed the most important animal in biomedical research. Here comes a very logical question. Why mice are so widely used in research? $98. And this is cheaper than my computer mouse. Mice are small, cheap, easy to maintain, and they also reproduce quickly, and they have a short lifespan of 2 to 3 years. That means in one experience you can observe several generations. But the main reason rats and mice are so widely used in medical testing is that their genetic, biological and behavioral characteristics closely resemble those of human, and many symptoms and conditions can be replicated in mice and rats. This is a comparison of DNA resemblance of a human and different animals. One more interesting fact that you could learn if you followed your biology lessons at school. You share only 50% of DNA with your mom and 50% of your DNA with your father. That means you way closer to a monkey and a banana than to your mother or your father. And here's a proof. Just look at this digital mirror. Let's understand what's behind these statistics. When you talk about math equations, it's easy to make comparisons. But when it comes to living things and their DNA, how can you say that this part of human equals this part of a monkey? And to answer this question, at first we have to understand what DNA is. If you have DNA, you have life. Congratulations! DNA is your blueprint. It's something your body uses to build your tissues, organs, muscles, everything. So if your DNA is bad, well, I'm sorry. DNA is contained in almost each cell of your organism, exceptions being red blood cells. They don't have DNA because they lose their nucleus as they mature to carry more oxygen and platelets. And also, sperm cells have only half of your DNA because they're designed to combine during reproduction to make a complete new set for a new organism. Not to fucking throw it away in a piece of toilet paper, fellas. Over 3 billion letters. When scientists analyze the DNA, they look at sequence of letters, like this. And for human, it's going to be over 3.2 billion. And each of your cells, even this small hair, will contain these 3 billion letters. To show you how to get from here to here, I decided to extract my DNA at home. I did not have all the chemicals at home and ran out of soap. So I decided to go to a drugstore. At first you need table salt and normal water. You make a solution by mixing them together. You stir it until the salt is completely dissolved in water. And to provide the solution with your DNA, you have then to wash your mouth with it. Without swallowing, of course. I didn't really like the taste of this one, it was disgusting to be honest.
Sweet berry wine!
You put it back to the glass, then you add a little bit of dish soap, as small as your dick. Stir it very carefully until fully dissolved. Then you add a chemical which is not easy to find. It's 90% alcohol, or as kids call it, isopropanol. And then you wait for 5 minutes. While waiting, you can already observe your DNA strands appearing in a glass and forming a shape. At the same time, my friend Harry decided to repeat the experience as well. If I had $2,000 more to spend for this episode, I would also do the sequencing of my DNA. Sequencing is when using chemicals you translate this stuff into letters. But let's just imagine that I have one and I got a big sequence of letters. Okay, now I have something I can work with. Let's get back to our question. How do you find similarities in DNA sequences? How can you say that this thing equals this? I bet you say something very intuitive, like let's compare it character by character. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We have 10 letters and we compare each one of them. So this is a match, 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 mismatch, match, match, and match. So we have 9 out of 10 of the letters which matched. That means we have 90% of the similarity between this DNA and this DNA. So this was how you can compare one DNA to another and get a proportion. Nucleotide, nucleotide, nucleotide. Do you really think scientists use this simple, straightforward method? They're probably smarter than you, but they actually do. And they call this single nucleotide polymorphism. Do you remember these statistics? This is exactly how they got it, with some small but huge modifications. Scientists were inserting and deleting the DNA in the way they felt, and somehow they produced an extremely high match between the monkey and the human DNA. Good job! And this is how they got this magical 98.8%. I believe some presidents know this method. And this is exactly how you become 60% similar to a banana. But what about these 50% of resemblance with your mother and your father? If you and I compared our DNA by looking at the letters in the sequence, we will get to 99.9% match. In all the people in the world, only a tiny small part of DNA is different. And this is where we get the number that humans are 99.9% similar. So even though you got 50% of your mom's DNA and 50% of your dad's DNA, those two sets are almost exactly the same. So finally you're way closer to your parents than to a monkey. Congratulations! There are actually other methods to compare the DNA strands, and they produce different results on the same sequences. However, if I start explaining them right now, I think I will lose your attention, so it may become the subject for another episode. One of these methods, the K-mer analysis, is extremely close to one of the analysis methods that we use in natural language processing, NLP. And this method is called n-grams. Okay, let's get back to the animals. I want to talk about humanized mice. With the technologies we have today, scientists may identify a part of the DNA, point to it, replace it with the sequence they want, or even make it non-functional. These genius guys introduced a knockout mouse back in 1989. A knockout mouse means some part of its DNA was modified, and in the way that it's not working anymore. So the normal protein generation is broken. Usually scientists do this to understand the importance of particular genes in different conditions. And there is also a knocking mouse. It's exactly the opposite. It's when some genes were modified or even introduced in the way that they are still working.
Niggas envy, blicky got the stiffy, got blicky, drumming.
And this is what scientists usually do to study some particular disorders. Actually, the humanized mice that were used to develop the COVID-19 vaccine were both knockouts and knock-ins. Fun fact! Before developing humanized mice for COVID research, by 2021, scientists had tried to infect a big bunch of animals with COVID and figured out that cats, monkeys, and hamsters can be naturally infected with COVID-19. I should have put a mask on my cat. There is a gene in human called ACE2. By the way, genes are just segments of DNA. A normal mouse cannot naturally be infected by COVID-19. So scientists created a GEM. GEM stands for genetically engineered mouse model. One of the ideal GEMs for COVID-19 research was considered to be a knockout knock-in design, in which the mouse ACE2 gene is deleted and the human ACE2 DNA is inserted in its place. I'm really impressed about where we got and what we can do with all these gene modification technologies. And while making this video, I learned a whole bunch of stuff I would love to talk about. Please, can you write down below whether you enjoyed the genetic topic or not? I will take your opinion into account to produce the new episode of State of the Old. Happy New Year!